my difficulty with the computer. I'm predominantly a Mac user these days. My name's Deborah. Thank you. I'll just stand in the middle so I can press the buttons. My name's Deborah. I'm a trainee in general surgery through the Royal Australasian College of Surgeons. And you very generously awarded me an oncology award, actually kind of towards the end of 2009. And I'd just like to explain what that allowed me to do and what's happened since then. So in 2009, I had been a surgical registrar for 5 years and was working steadily through the somewhat prolonged training process to become eventually a consultant general surgeon. And the opportunity arose for me to step sideways from my clinical training to undertake a period of research, that being a PhD through the University of Auckland, looking at the genomics, the underlying genetic changes of colorectal cancer. Now that's an expensive thing to do, and it's expensive on two fronts. Number one, I'm quite expensive now. More so, not as much as you, but more so perhaps than I would have been had I been 21 and just finishing my undergraduate degree. The second thing to acknowledge is that this kind of molecular biological research is extraordinarily expensive to undertake. And so you need to really think about some quite serious levels of funding in order to be able to do this kind of thing. You can't do it alone, you have to have the support of an extremely good research group. And that's who I'd like to acknowledge right at the beginning of my talk. People you see behind me are on your right, Chris Print at the top, and Aaron Mary below him, and they're my supervisors. Aaron is a colorectal surgeon at Auckland Hospital. He did his own PhD in this field, finishing about 10 years ago in Otago. And Chris is the scientist with a clinical background in the molecular medicine and pathology department. So I'm actually going to see them later on this afternoon. These are my mentors, my supervisors, my academic and to a degree clinical overseers and my mentors. On the other side of the slide, you see the rest of our research group. Just to quickly introduce them: Mitsu, Annette, Cherie, Daniel, Sonali. They chose some of their own pictures. And I really can't talk about too much. I can't overemphasize how important it is the people that surround you. You can't do any of this on your own. It's very important, it's very complicated, it's very expensive, it needs a lot of equipment, and it also needs a lot of knowledge. You all know there's so much you can read in a textbook, but for the Gestalt experience aspects of things, you need people like these who are incredibly generous with their knowledge and with their time, and have allowed me to do everything I've done so far. So this is a little bit, this slide introduces the idea of what our area of research is about. This slide introduces colorectal cancer, which is the cancer I am particularly working on. The things I'm going to say are true for most, if not all, cancers. And that is the idea that a cancer arises through a series of progressive and interlinked genetic changes within a cell that allow it to take on certain properties which normal cells do not have. Allow it to live longer, to be less subject to the normal cell turnover in that area, to be able to invade and spread to other organs. We now understand that that process is not a linear, stepwise progression, but more complicated than that, involving a number of changes and changes which interrelate to each other. So that is the basis of the kind of research that we do. The first thing that I did was a survey. There are a number of tools available internationally and in New Zealand which are expanding the idea that not all cancers are the same. So in this instance, not all bowel cancers are the same. Because although you get to the same endpoint, the genetic and genomic changes which take place in that individual's body to reach that endpoint are not the same. And there is an international movement to try and tailor cancer treatment, and indeed treatment for other diseases, to its occurrence in that particular individual. We call this the personalisation of healthcare. Now, there are a number of tools available in New Zealand which are designed to help doctors individualise the care or personalise the care that they give to an individual patient. These can be in the form of computer programs which allow you to put in certain characteristics of the patient in front of you. give you information about, for example, the likelihood that they will respond to chemotherapy, the likelihood that they will have disease relapse. There's another group of tools which are molecular tests, which are looking at particular tumour biology of that particular individual's particular tumour, and again gives you information, for example, about their likelihood of recurrence. Will they respond to this particular treatment, and so on. So we know that these tools are out there. And what we wanted to start off by doing is ask, are they used in New Zealand? What are they used for? How much? Do they actually change what happens? Do they change what doctors do? And so that's where I started from. And I've got two pie charts behind me. On the right of your picture, left of your picture, sorry, is talking about those computerized tools, and on the right about molecular tests. So what I've said here, of half of the people who responded to my survey, and all of the respondents were doctors who treat cancer in New Zealand, over half of them currently use prognostic tools, those computerized tests, when they're treating patients with cancer. And a third of them change what they do on the basis of those results. If we look over here to molecular tests, people who use them are the blue segment, used by fewer, and that isn't surprising. Some of these tests are extremely expensive, many aren't funded in the health service, but the likelihood that the result of it will change what they do is higher. So essentially we can conclude that these really already have a place in the New Zealand management of cancer. That comes out absolutely terribly, I apologise for that. The next thing we asked the respondents to our survey is, what do you think is going to happen over the next 10 years? Do you think these are going to have more influence and impact on what you do for your patients? We asked, do you think you'll use them more frequently or less? Do you think that you'll change your decisions more on the basis of them? Do you think they'll be better supported by evidence? And the big pale blue wedge is yes. So you can see, over 90% of our respondents said yes, every one of those questions. These are going to be really important things. Brian's giving me the hurry up, I'll hurry up. So it's inherent on people like me, or like us, to make sure that their faith in these tests is supported by a really good evidence demonstrating that they work. And we argue that they may well do, but currently there isn't necessarily enough evidence to demonstrate that. So what are we doing in the lab? Well, this is just a quick demonstration of some of the things we're doing, and again I acknowledge the research group as part of this. These are colorectal cancer cells. It's not what they look like in us. They're grown artificially as a cell culture environment. The ones on the left are being grown nice and gently, and the ones on the right, they're brothers and sisters grown in exactly the same environment, except we've put chemotherapy on them. And I think that you can see they don't really look very happy. So we can see with the microscope they don't look happy. But we've taken the genetic material out of these cells and run them on a type of technology called a microarray chip to show us the genetic changes that happen in that process. And this is the kind of data that we get out, or that we can transform it into. This is another experiment by my colleague Sonali, who's looking at what happens to these cells when you expose them to a very low oxygen environment. So imagine if you have a tumour which is a lump. The middle of that tumour will quite often have a poor blood supply, low oxygen, and she's looking at the changes which happen there. This is to remind me, your funding allowed me to make that transition from being, as you see me on that brochure, surgical registrar in the operating theatre, to spending this time in the lab. And that was great for the first year, but what next? Well, this reminds me what next. It's quite difficult when you make that transition because you don't have any, I have done research before, but nothing like this. And so your faith in me allowed me to make that step, along with the support of my college, the Royal Australasian College of Surgeons. At the end of last year, I was absolutely delighted. I have to say, I think my supervisor was kind of surprised. I've got $250,000 worth of funding from the Health Research Council, which will support support me for the next 3 years. So that's really fantastic. So that means that your $10,000 has become $250,000. So thank you very much.